Good morning, everybody. So until last class, we covered some basic textbook topics in computational neuroscience. Now, since we have this new issue now that now so some of the classmates cannot join because of placements. I thought I'll cover some research topics more and not the regular textbook stuff. So I'll talk about some of the work we've been doing in my lab. So I'll start with this topic of modeling the basal ganglia, which is an important circuit deep inside the brain. It's a subcortical circuit. So we've been working on this for the last almost 15 years, and I'll just highlight a few main ideas from this work. So as you know, one of the recurrent themes in the course and also the kind of modeling approach I keep saying that should be given more emphasis is not just to model details and model, develop like curve fitting type models, but try to go to the fundamental principles and then develop models that reflect the fundamental principles and not just curve fitting type models. So a lot of people are saying that neuroscience currently is like the stage of astronomy, say 500 years ago or before the time of Newton. So before time of Newton, even before Kepler, it was mostly observational astronomy, and people would just fit some curves to their observations. So for example, planetary motion was explained using epicycles and things like that. So multiple cycles superimposed on cycles. But because people used to think that Earth is the center of the universe and all planets go around the Earth. So so because of that assumption, the planetary orbits looked very complicated. But once you assume that or understood that Sun is the center of the solar system and planets move around that, and with Kepler's laws, we learned that planets move in elliptical orbits with Sun as foci, Sun as the focus. Then things looked much simpler. All the data looked very simple, and you don't need any epicycles. And that led to fundamental principles. So that it led to the so-called Newtonian revolution, which has shown that the universal law of gravitation applies not only to our solar system, but it applies practically anywhere, any solar system anywhere in the universe. That's why it's called universal law of gravitation. So that is the flavor of true science, right? It will become universal. You don't just try to fit observation data and come up with a different model for every observation. So we are not yet there in neuroscience. That's why you have such a explosion of theories and models and all that, because we're still groping in the dark. Okay, the situation with basal ganglia is no different. I'll explain in a minute. So the basal ganglia is the deep brain system, a circuit. It's a subcortical circuit. Brain has this surface of tissue which consists of 6 layers of neurons. The sheet of tissue called the cortex. And so everything else that is inside the brain is called the subcortex. What is there below the cortex? And you have these lot of subcortical structures, like thalamus and hippocampus and all these things. One such important, it's not a structure, it's a circuit, is called the basal ganglia. So you can see them here in the left picture. It's in the coronal section. But you see that yellow and blue and all that brown. These are all a bunch of masses of neurons. They're called the nuclei. And that's a terminology in neuroanatomy. A bunch of neurons is called the nucleus. And they're all connected very tightly, so they form a system. And this system is called the basal ganglia. So ganglia is a general term again for a mass of neurons. It's called basal because it's at the base of the brain. It's way below, right? So the base of the brain. Therefore, all these ganglia together is constitute what is called the basal ganglia. It has many nuclei in it, and like for example, the globus pallidus and striatum, and subthalamic nucleus, and all that. They're all connected in some fashion. So let us not look at these anatomical pictures because they look very confusing. It's very hard to make out which is connected to which area and say things like that. So let us look at a more block diagram, which is the kind of picture that an engineer would like to draw. Right. So the basal ganglia architecture is like this. It gets input from the cortex. Right. So they see these main areas of basal ganglia like striatum, and GPI stands for globus pallidus interna. SNR stands for substantia nigra pars reticulata, and GPE stands for globus pallidus externa, and STN stands for subthalamic nucleus. So don't worry about this terminology too much. Just you can even think of them as XYZ if you like. But basically, it's a bunch of nuclei connected, by some pathways. This whole system gets input from the cortex. So you can see that at the top. And then that information goes through a bunch of pathways within the basal ganglia. And the input port of the basal ganglia is the striatum, that's the blue box right there at the slightly at the top. And then it goes via two pathways. So the output port is GPA and SNR. There are two output ports. So there's a short pathway connecting the striatum and the GPA/SNR, and there's a longer pathway that goes from striatum to GPE to STN to GPI. Right. So this shorter pathway is called the direct pathway, and the longer pathway going from striatum to GPE to STN to GPI is called the indirect pathway. Okay, so information from cortex comes to the basal ganglia. It goes through two pathways within the basal ganglia. And output of the basal ganglia from GPA to SNR goes to thalamus, which you know is a major sensory motor hub in the brain. And from thalamus, it goes back to the cortex. So input from the cortex and return output back to the cortex. The only thing is the input comes from one area and output goes to a different area of the cortex. That's a different matter. I'll come to that issue later. Now, so this system, right, why is it important? What does it do? So it does a lot of functions. In fact, if you divide all brain functions into four major domains, sensory motor function, that is all your senses, right, and your motor output, right, that is sensory motor function. Then cognitive function, that is your memory and decision making, planning, things like that. Then you have autonomic affective function, that is emotions, right, the functions of what is called the limbic system, which is again a big circuit in the brain, right, your depression and mood and things like that. And then finally, your autonomic function, that is how brain controls the internal organs, the viscera inside the body. So this is all that the brain does. Whatever brain does can be put in one of these four compartments. Now, basal ganglia has a role in all the four major domains. So therefore, if there's any damage in basal ganglia, you will see some kind of a problem in all these four major domains. So specifically, it is involved in action selection. That is, if I have multiple actions that are presenting themselves, and which one will I choose in a given context, right? You can also call it decision making, right? And basal ganglia has a role in that. Sequence generation, how do I memorize sequences and produce sequences? So for example, a complex dance movement or a complex movement in some martial arts, right? How do I produce them? So basal ganglia has a role in that. And if basal ganglia damage occurs, then you will have problem with that kind of a function. Right. So similarly, action gating and reward-based learning. So that is, we often learn by reward. I mean, why have we taken this course? Okay, one is this pure love of knowledge. That is one reason. Other is, getting a good grade, hopefully, and getting some, admission in some good university and higher studies, and so that's a reward, right? So we do things for reward. We do learn for the sake of reward. So that is reward-based learning. So how does it work? How does brain conduct all that? Then timing. So basal ganglia is involved in, so brain does a lot of operations which involve timing with the activities which are spread out over a duration, a time duration. And here there are three different levels of scales of timing. It operates at three different time scales. For fast kinds of movements or activities, it is cerebellum that is involved, right? That's called millisecond timing. Then for slightly longer durations, running into seconds to minutes, that is called interval timing, and this is where basal ganglia is involved. For activities which run over, stretch over longer durations, like hours to days and all that, you have a different brain system called the suprachiasmatic nucleus. So the timing activities of the brain are conducted by three different systems, and basal ganglia is involved in one of them, the interval time. Okay, then exploratory behavior and working memory, and so you can give a lot of functions. Maybe there is a more logical way of grouping the functions is based on the four domains of brain function. Right, and so it's a very important influential system, so it's important to study that. And for a long time people were really puzzled by the basal ganglia's function, because how can the same circuit have influence over such wide variety of human brain functions, right? Into this famous neurologist from British neurologist Marsden, he referred to the mysterious motor functions of basal ganglia. I mean, for example, basal ganglia damage will lead to, even in motor function alone, right, a wide range of impairments, which I'll list out later. So it's a big problem to understand how basal ganglia works. Now, people have been proposing computation models. So, for example, and these are very old papers. I mean, this it's now like there's so much literature coming out in basal ganglia modeling area these days. But these are some of the older papers. Like, for example, people have been proposing models for different functions of basal ganglia. So that there are people who only highlight the reward-based learning aspect of basal ganglia, or there are models of sequence generation. How do the basal ganglia generate sequences? Or there are models of working memory. So how does basal ganglia remember something for a short while and use that information for decision making? Or how does basal ganglia control or conduct timing activities over a scale of seconds to minutes? Or action gating, dimension reduction. So the different models propose to different functions of basal ganglia. But the problem is the situation at that time, like this was in 2005 when we just got into this whole area. The situation was like that, and it's like this blind monks trying to figure out what an elephant looks like. Right? Some monks were looking at only the legs and they thought it's like pillars, and some monks were looking at the trunk and they thought it's like a snake, and etc. etc. So each person was looking at one aspect of the elephant. They didn't have an integrated knowledge of what the complete animal looks like. So one reason why we are interested in basal ganglia from an applied point of view is that it is related to an important disease called an incurable disease right now. It's an unsolved problem called the Parkinson's disease or PD. Okay, it is a kind of a disorder called a neurodegenerative disorder. So where there is a progressive loss of cells in a region of basal ganglia called the substantia nigra pars compacta, or SNC, which is located right here. This yellow box towards the right with this dotted lines coming out of it. So this part of the brain or basal ganglia has cells which release a substance called dopamine. And because these cells are dying for some reason, dopamine levels go down in the brain. And because of that, we have lots of problems arising. Now, we don't know why these cells are dying, although there's some theories, and even we have worked out a computational theory with a PhD thesis, which the student has just completed thesis a few months ago. So, but still, and as of date, we don't have a comprehensive theory, and we don't have a well accepted theory, right? And about why these cells are dying. But once they start dying, there is a continuous loss of cells, and you can't stop it. And that is why it's called a neurodegenerative disorder. Once the degeneration starts, there is no stopping. You can only manage it, but you cannot completely arrest the loss of cells. Now, this disease is characterized by four major symptoms. So these are called the cardinal symptoms, the most important symptoms. That's a tremor, that is shaking of limbs, usually upper extremities, the hands, and therefore it was called shaking palsy because it has a strange combination of rigidity and tremor. Okay, so you also get rigid joints, right? And postural abnormalities. These people typically walk with a little stoop, and even when they walk, they take short shuffling steps called fascination. Then they also tend to move slowly, which is called bradykinesia. Brady is slow and kinesia is movement. So all the four relate to movement problems or motor function problems. But like I said, damage to basal ganglia can manifest as impairment in all the four major domains. Even cognition is affected. In fact, not only bradykinesia, these patients also have bradyphrenia, that is slowness of thought. So it affects cognition pretty deeply. And if you look at motor functions alone, they have problems in reaching, in handwriting, in eye movements, speech, and posture, etc. Just a minute, I'll come back in a second. Just a small problem. Hindu. Stop for a second. Hello. Yeah, hi. So like I said, in motor function alone, you have so many different modalities, like the reaching movements. How do you reach to a target? Or handwriting, right? Or movements of eyes. Speech, which is the movements of the articulatory apparatus, your larynx and jaws and all that, tongue and so on. Then posture, right? All these are affected. So pretty much there's nothing left. In all movement can be affected in principle because of damage to SNC. At a given patient, you may not see all the problems, but in principle, all these are possible. You will see some of these in some patients, in PD patients. So on the right bottom, you see this normal handwriting versus PD handwriting. So this is a Tamil word called Sri Ramajayam. The upper one is written by a normal person, elderly person, but a normal person. The lower ones are written by PD patient, age matched patient. Okay, so you see that they write very small letters, which is called micrographia, that is small writing. And also you see the letters are not very clear, not very distinct. So it's important to study basal ganglia for two reasons, for academic reasons and basic science reasons, and also for practical applied reasons. Okay, so now the question is, how does the basal ganglia work, right? How does it, what is the function? So let us start with the classical picture, which is what you will see if you take a textbook on neurology or the clinical neuroscience. So the way the story goes is, we know that you have these two pathways in basal ganglia, the direct pathway and indirect pathway. So here the neurotransmitters are, from striatum to GPI, you have GABA, so it's inhibitory stage. That is shown by this minus sign here. From GPA or beyond, so the output of GPA goes to thalamus. There again you have an inhibitory stage. That's why again you have a minus, right? And if you look at the indirect pathway, you have again a GABA, then again glutamate, which is positive, then negative GABA. So you see that along the direct pathway stage. So if you look at the pathway from striatum to thalamus is not shown here, but the output of GPA goes to thalamus. You have two inhibitory stages. So two negatives make a positive. So effectively, that's like a positive pathway, all right? And then if you look at indirect pathway, you have inhibitory, and then excitatory, inhibitory. So an odd number of inhibitory stages. So that is on the whole inhibitory. So therefore, the idea is if you activate the direct pathway, it'll have a positive effect on movement. It'll facilitate movement. It'll release movement. And if you activate the indirect pathway, it will inhibit the movement or it'll stop movement. Okay, so what determines which pathway is selected, which is activated? So there the story goes like this. So the SNC neurons project onto the striatum, which is actually a big nucleus, and release dopamine onto striatum. So within the striatum, there is a cellular machinery. I'll elaborate that later on, right? Where because of the action of the dopamine on these cells of striatum, the signals coming from the cortex, so input to the striatum comes from the cortex, direct input from cortex. So the signals coming from striatum, depending upon the dopamine level in striatum, will either go through straight along the direct pathway or take a kind of a detour and go along the indirect pathway. So now at high dopamine level, signal goes straight along the direct pathway. At low dopamine level, signal take a reroute, like a detour, and go along the indirect pathway. So therefore, so the direct pathway is also called a go pathway because when you activate the direct pathway, the movement gets facilitated. You get to go. Similarly, when you activate the indirect pathway, the movement is inhibited. So it's called. It's also called the no-go pathway. So now we can summarize that at high dopamine levels in striatum, right, the direct pathway is selected. Therefore, the subject gets to go, movement is facilitated. At low dopamine levels, indirect pathway is selected. Therefore, the movement is inhibited. The subject cannot go, cannot move. Okay, so that's summarized in this box in the right bottom. High dopamine, go, low dopamine, no go. So therefore, in case of PD or Parkinson's disease, your dopamine levels are less because these cells which release dopamine are dying. So therefore, you cannot move. You have problem moving, and so that's how the situation is summarized or explained, right? In case of PD. So this is what you will see in older papers. I know there's a paper by Alvin et al. From 1989, or older textbooks. The thing is, the real story is a bit more complicated than that. So to understand that, you should look at a different aspect of this chemical called dopamine. Now, dopamine is not just, its a function doesn't stop with just controlling the switching between these two pathways. Dopamine is called brain's pleasure chemical. This is a chemical that gets released by the dopamine cells. There are other clusters other than SNC. There's another cluster called the VTA, or ventral tegmental area. Both these are main clusters of cells which release dopamine. So whenever you experience pleasure, right, these cells fire and they release dopamine to various targets in the brain. So the activity of these cells indicates that you are happy. So therefore, specifically, dopamine release is triggered by brain stimulation. So specifically, what that means is, like, there are certain areas in the brain which, when stimulated, gives you a sense of pleasure, right? It's like a completely unconditional pleasure, right? Normally, when you talk about pleasure, you need to have some support. You need to have something that makes you happy. You need an object of pleasure. But there are certain brain areas where if you just put an electrode and activate them, electrically activate them, stimulate them, you just feel pleasure. So these kinds of centers were known for a long time, and even some early experiments on these electrical activation studies, brain stimulation studies. So animals were rigged up with this kind of electrodes put inside these pleasure centers of the brain, and they had an option to just press a lever and stimulate themselves. So they would keep on stimulating themselves even at the cost of foregoing food. They would literally starve themselves, but they'll keep on stimulating themselves, right? And so that's how strong these centers are. And it is generally now known that these are the kinds of centers which are activated when people use drugs. That's why people, have such addiction, craving for drugs, because they activate these pleasure centers of the brain, right? So what happens is, when these centers are activated, dopamine is released. That's psychomotor stimulants and opiates and all that. Even food is not drugs, but food makes you happy, right? And we like to eat food. So especially tasty food, gourmet, food. So that's because say this such a food stimuli also result in release of dopamine. Now, contrarily, if you give drugs which counteract the effect of dopamine, like dopamine antagonists, these are chemicals which counteract the effect of dopamine. Such chemicals attenuate the pleasurable experience of above. Right, whatever pleasures you have had because of dopamine release, that pleasure will simply vanish and evaporate if you also take a dopamine antagonist, another drug which will counteract dopamine. So basically, your happiness is all just a bunch of chemistry. That's all. So similarly, even if you presentation of beautiful faces, that's why we like to watch stars, like in movies who are good looking, right? Our images of lovers and monetary rewards. So we like to get cash because, again, cash rewards also activate the same reward centers in the brain. So all these stimuli trigger the dopamine reward system. So dopamine gets released, and that will act on other brain areas, primarily basal ganglia, in the current context. So there is a whole reward system in the brain where the dopamine plays a key role, and so that's what represents to you, to the brain, that you are happy and there is something pleasurable going on. Okay, so that's a major finding, because once you have something in the brain that will signal reward, you can do a lot of things with it. You can learn a lot of things with it, because a lot of learning is related to reward, right? Reward is what decides whether you want to learn something or not learn something. But we'll come to that in a minute. Now, that's a good starting point. In addition, people went further and found that what the dopamine cells, the activity of dopamine cells represents, is not just a reward, but something more subtle than that. So these experiments were done by Wolfram Schultz and his colleagues, earliest paper from 1997. So in this experiment, there is a monkey, and the monkey, there's a empty box in front of the monkey. A box has a trap door. Monkey gets to put its hand inside the box, right, and inside the box there'll be a lever, there'll be a wire to which you'll have a piece of apple hanging. Okay, there's a piece of apple stuck to that wire, and then monkey can put its hand in the box and grab the apple. Now so there were electrodes inserted in the ventral tegmental area of the monkey, which is another dopamine center, dopamine clusters of cells in the brain. Now, so in the early trials, you see that the top panel. So you see there is a dark trace in the top that represents average firing of a whole bunch of cells in VTA. And those dotted lines, it looks like Morse code on the bottom, is each dot represents a firing event. So if you think of them as parallel lines of dots, each parallel line represents activity of one neuron, and each dot in a given line represents the time at which that neuron fired. Now you average all the activities of those neurons and plot them as one average summary plot. That's what you see in the very top of each panel. So now we see that letter R in the top panel that represents the time at which the reward was given. So in this case, the reward is that piece of apple. So when the animal touches that piece of apple, the monkey's brain interprets that as reward, and therefore the cells immediately fire. So therefore, you can see that the firing rate goes up immediately after that reward instant. So this is a familiar thing. We already know this. So basically, what we knew at level of behavior is now confirmed at the level of cells, right? We knew that dopamine gets released, and that's in response to pleasurable stimuli. So now that these guys are just observing that at level of single cells, because when the monkey touches the apple, that these cells start firing, indicating that apple is a good thing because it's a source of pleasure or food. But look at the second set of trials. In the second set of trials, just before the monkey gets to touch the apple, there is another stimulus given. It's a conditioning stimulus. We talked about conditioning stimulus and all that in Pavlovian conditioning, right, or classical conditioning, I think a couple of classes ago. So here, conditioning stimulus is a kind of a flash of light. So they give a flash of light followed by a small delay, at which time the monkey gets to stretch its hand and touch the apple. And remember that the apple is hidden. It has to put its hand inside a closed box and then grab the apple. So as it keeps doing this, that is this flash of light and it touches the apple, flash of light and touches the apple. So after doing this kind of a pairing, right, a few times, the monkey kind of understood that moment it sees the flash of light, it gets to grab the apple. So what happened was after a bunch of these trials, the neurons started firing right when the monkey saw this flash of light. So you see that increased firing right at the time of conditioning stimulus, which is a flash of light. And after a delay, when the monkey actually gets to grab the apple, there's no change in firing. So this trial indicates that these neurons are representing a prediction of reward and not the actual reward. Actual reward came a bit later when it touched the apple. But when the flash of light came, it's that event is predicting the arrival of reward a bit later. So neurons seem to be firing to represent that. That's what second set of trials seem to indicate. In the third set of trials, actually towards the end of this whole experiment, they did one more thing. There was a flash of light. In response to that, the neurons fired like before. But at the time when the animal actually gets to put its hand in the box and touch the apple, they cheated the monkey and just allowed the box to be empty. So when the animal put its hand, there was nothing inside. In such a trial, the firing rate of these neurons went down than even normal. Okay, so when there is actually reward and the reward is expected, there's no change in firing rate. But when it is expecting a reward but there's no reward in the box, then the firing rate actually went down. So this last trial indicated that what these neurons are representing is not just a predicted reward, but some kind of an error between the actual prediction, the prediction of the reward and the actual reward. So therefore, the dopamine activity of these cells, the BTS cells, seem to indicate the total actual future reward minus total expected future reward. So some kind of a reward-related error, what is called the reward prediction error or RPE. Now, there's a lot of controversy about this. It's not always dependent on RP. It is not always representing RP. It is sometimes it's representing something else and so on. But this early experiment by Schulz et al., that's what it has indicated. Now that had led to a lot of interesting theory building of basal ganglia function. So one such thing is called reinforcement learning. One such theory which has inspired modeling of basal ganglia is what is called reinforcement learning, or learning by rewards, right, or reinforcements. So the basic idea of reinforcement learning is like this, right? And so a lot of learning basically involves learning to relate a stimulus and a response. That is, for a given stimulus, what is the correct response? What do you mean by correct response? That is a response which will give you a reward. Because for a given stimulus, if I produce a response and that response leads to reward, then you will figure out, you'll understand that that response is correct for that stimulus, and you will strengthen the connection between that stimulus and the response, so that next time around, when you see the same stimulus, you will produce the same response. Because therefore that stimulus response relationship will be reinforced if you get a positive reward. So similarly, or contrarily, for a given stimulus, you produce a response and you get a punishment, some negative reward, then you understand that that stimulus response relationship is wrong, and you will try to suppress that connection, or you will attenuate that connection. So basically, you can, in this intuitive picture, you can see that your rewards and punishment that you get from the world, from the environment, will let you modulate the connection, the relationship between stimulus and response. So this is called reinforcement learning. So you can see that anatomically speaking, basal ganglia is well positioned to perform this role of reinforcement learning, because if you look at the connections that basal ganglia receives, it receives extensively from the cortex, from both motor and sensory cortices. Okay, so I'm just showing that using this thick block arrow. And so the top rectangle is sensory motor cortex. Actually, the sensory cortex on the right, projecting to motor cortex. And input to this whole cortex is sensory stimulus, output is motor response. Now, this whole sensory motor cortex sends projections to basal ganglia, which is known anatomically. And it also receives input from SNC, which represents reward. So you see that basal ganglia knows what you have done before and what was the stimulus to you when you did that before. And that response did give you reward or punishment. That also it knows, because it gets input from SNC. It knows this, whether dopamine level is high or low. High means reward and low means punishment. Let me put it that way. I'm making the story simple, but let us stick with that for now. So by combining these three pieces of information, it will decide whether what you have done just now is good or bad, and make that recommendation to the motor cortex. Because output of the basal ganglia doesn't go to sensory cortex; it goes mainly to motor cortex. Actually, the motor and even prefrontal areas where you have regions which are involved in decision making, planning, and all that. So it informs them as to what is a good action to perform. So based on that, the sensory motor cortical pathways get adjusted, get adapted, and then the cortex slowly adjusts itself, learns what is the correct thing to do, how to respond correctly and optimally in terms of what kind of actions produce high reward. So to illustrate this, let us build on this idea a little bit more. Let us say there's a monkey which is presented with two choices. One is to press a yellow button, another is to press a red button. Now it turns out that the yellow button gives it reward, which is in form of like orange juice. Because that's the kind of reward that people normally give in experiments. Or if it presses the red button, it gets a punishment in the form of an electric shock, which is typically the kind of punishment that animals get in experiments. But initially, the animal doesn't know that. It doesn't know what yellow means and what red means. Okay, so let us say this yellow bar and red bar that you see here represents the kind of weightage the animal has in its mind, in its brain, about what is more rewarding. Initially, it thinks both are equally good, so you have both these bars are equally high. Then it presses the yellow button, it gets orange juice, so it got reward. So now it understands that yellow is a good thing. So its weightage for yellow goes up. So similarly, next it tries the red button. It gets a shock, right? And then understands that red means punishment or negative reward. So its weightage for red goes down. So what we call weightages are more formally called values in reinforcement learning literature. So yellow has high value and red has low value. Now, once the animal learns these values of different actions, it uses these values from now on to decide which action is a good action, and then it uses these values to decide these actions. Okay, so basically the policy now is choose actions which have high value. Don't choose actions which have low value, which is common sense. But to come up with a comprehensive decision-making system, you need a third element, which is the ability to try out unknown actions. Okay, so you know some actions which are good, have high value, you know some actions which are bad, have low value, but there could be actions which you have never tried out. You don't know whether they are good or bad, so you need to be able to try it out. Only then you can ascertain what their value is. So this ability to try out unknown actions and ascertain their corresponding value, it's called exploration. So let me give an example. Suppose you go to restaurants, right? I mean, this is because of COVID, you don't go out much, but normally we like to go to restaurants. And typically, there are some restaurants we always go. We like to go because you always get good food there. Which means we have tried food there before. Therefore, these restaurants have high value. And some restaurants we know that they are bad because we went there before and we had bad experiences. Okay, so if that is your whole approach to restaurant going, that's not very optimal because there will be new restaurants which have never heard of or never sampled, right? And they might be better than the best restaurants you have already seen. So unless you try out those restaurants, risk it a little bit, try out those restaurants, you will never, obtain that reward that such restaurants can potentially give you. So that feature of ability to try out is called exploration. So to summarize, to come up with a comprehensive decision-making system, you need to have the system should have the following things. It needs to have a reward signal. It needs to be able to compute and generate values, right? It needs to be able to explore new actions which it has never performed, so that right is always up to date and it knows, because unless you explore new actions, you won't be able to keep up with the changing conditions of the external environment. So you need to have exploration. So then you have actions. You need to perform some actions, like for example, choosing yellow button versus red button. Then you need to have some kind of a state. So in the previous example, I didn't really talk about state, but state is the input that you get from the sensory cortex, your sensory state. Now that is what these are the elements of a reinforcement learning system. But how does the Basal ganglia do all this? What are the counterparts? What are the substrates of all these reinforcement learning components or concepts in the basal ganglia anatomy? Now, certain things are easy to work out. For example, dopamine cell activity represents reward or reward prediction error, more specifically, but it's something to do with reward, so that we know. Now, what about values? Where are values located? So it turns out that values are computed in striatum because there is a lot of evidence from functional imaging. So in these experiments, subjects are asked to play some gambling games, just like what we have done with this yellow button, red button. In gambling, you make multiple choices, and based on choice, you get some money or lose money. So then, based on that experience, right, you understand that certain choices are good, certain choices are bad, and keep on trying to adjust your choices so that you maximize your money that you make, right, your returns. So that knowledge seems to be stored in striatum. There's a lot of evidence from functional imaging. That's where the value information is stored. Now the state information comes from sensory cortex, so that is coming as an input to the striatum. Now this exploration business, where is it occurring? Exploration means ability to try out actions somewhat randomly, right? Where if basal ganglia is to be thought of as the reinforcement learning engine, then where in the basal ganglia is this exploration coming from? So that was the question, and when we got into this field, we didn't find a good answer to this. So the classical picture simply says the direct pathway is go and the indirect pathway is no go. But where is exploration happening? Okay, so here we took a very interesting, important detour, because one thing that the classical stories ignore is that when they say the indirect pathway simply is a no-go pathway, I mean they try to rationalize that in terms of the number of inhibitory stages, right? And that the number of inhibitory stages on the indirect pathway is an odd number. It's three, right? Negative, negative, positive, negative. Number of inhibitory is three, therefore it is on the whole considered inhibitory, but it's not as simple as that, because if you look at the indirect pathway, it is not a feed-forward system, right? Only when you have feed-forward system, you can count this number of inhibitions and say it is on the whole inhibitory or on the whole excitatory and things like that. But it has a feedback, here a branch coming from STN back to GPE. So if a STN GPE will form a feed forward feedback loop, so therefore the simple arguments in terms of the odd number and even number of inhibitors, etc., are not valid anymore. And moment you have a loop, we know that you get very complex dynamics, right? And so therefore, and the feedback, interestingly, happens to be a glutamate feedback. So feed forward from GPE to STN is negative, that's GABA. But feedback from STN to GPE is glutamate, that is excitation. So now the STN GPE can have, in principle, complex dynamics. Okay, I'll say more about that soon. So we did some studies on that and kind of hypothesized that the exploration is coming out of the STN-GP loop or this indirect pathway. So on the whole, if you look at, we need to expand the classical picture of basal ganglia function. So the direct pathway is still the go pathway, because if you activate that, you facilitate movement. But the indirect pathway we proposed is not only the go pathway, it is also the explore pathway. So it does exploration also. This is where the ability to try out different actions is coming from. And why is that? I'll explain in a second. So this is our summary. And also we showed using simulations that for high dopamine levels, you get to choose the go pathway. For very low dopamine levels, you get to choose the no-go regime. It's not the pathway, it's no-go regime. That is, you have inhibition of movement. But for intermediate levels of dopamine, it's not neither go nor no-go. It is explore. That is, you get to choose, but not just the highest value actions, but other actions in a somewhat random fashion. So we said that this is what happens at intermediate levels of dopamine, and the indirect pathway is the one which will enable that kind of a choice, right? And so we said we need to expand the classical model. Now, how do you justify that? How do you say that the indirect pathway is what is doing the exploration? So here again, let us visit some of the traditional pictures of the function of SCNGP or function of indirect pathway. So the SCNGP as a no-go pathway has been known for a long time. A lot of people have said that. And then another function they said is called focusing and sequencing and action selection and so on and so forth. So I won't go into these theories. But if you look at modeling results, SNGP system forms a loop. It's excitatory inhibitory loop. That is, you have excitatory nucleus, which is STN, connected to inhibitory nucleus, which is GPE, and these two form a loop. And it is known from very long time in modeling literature that if you have a excitatory cluster of neurons connected to inhibitory cluster of neurons with back and forth connections, such a system can produce oscillatory activity. This has been known for a long time, and a lot of people have done models of this kind. In addition, this second paper, you see the Terman et al. 2002, they have proposed a model of SC and GP coupled together, and that model has shown that in such conditions, the network can show complex spiking, right? Complex spiking activity and traveling waves and synchronized bursts and things like that. So it can show very rich dynamics because it's connected as a loop. So that's some results coming from modeling literature. Now let us look at experimental literature. So we're trying to make a case to say that SNGP is probably the part of the basal ganglia which is involved in exploration. Now, it is known from experimental studies, if you look at the first sentence, decorticated kittens show exploration. That is, it is possible to create an animal preparation where the cortex is completely removed and the animal will get to move and survive completely based on the subcortical circuitry. So even such kittens were able to show exploratory activity. That is, they are able to search for some food, right? That is exploration. Now, how is it possible if there is no machinery for exploration somewhere in the subcortex? But that's somewhat generic, somewhat non-specific. But let us see the second result. STN lesions cause perseverative behavior. Now, perseverative behavior is a psychological term. What this means is people who perform the same action, even though that action is known not to give reward. Okay, so normally you try out an intelligent person, right? Tries out multiple actions, and actions which give reward, you stick with them, and actions which don't give reward, you drop them. But if you keep on performing actions, even though you know from past experience that they don't give reward, that is called perseveration. Now, STN lesions cause such behavior. And perseveration is obviously an opposite of exploration. Exploration means trying out things, different things, not getting stuck on one action, even though you know that it's not giving you a reward. So STN lesion can show such behavior, so which kind of indicates that STN-GP system contributes to exploratory behavior. Now that is still at behavioral level. Now what seem to really indicate that STN-GP is probably giving or contributing to exploration is at the level of electrophysiology. That is, if you insert electrodes in STN and GP and record the activity, what people found is under dopamine deficient conditions, like what happens in Parkinson's, the firing rates haven't changed much in STN-GP, but the activity is slightly more regular. There's reduced complexity, there's higher correlation among the activities of STN-GP neurons. Now, so thing is, when you want the animal to perform different actions randomly, what kind of neural activity do you think can give you that kind of behavior? So you can intuitively guess that only when neural activity has sufficient dynamic complexity, right? So that means for the same stimulus, if the dynamics of that system can have sufficient complexity, it will produce different kinds of dynamics even though the input is the same. When the system has that kind of behavior, then it can, in principle, drive exploratory behavior because there is spontaneous variation in the response. It doesn't have to be informed by the input. Okay, so any part of the brain which is involved in exploration should have some level of dynamic complexity. So you see that happening in STN-GP, and you see a loss of that complexity under dopamine deficient conditions, which is like the disease condition in Parkinson's disease or PD conditions. So that is the background. So with this background in mind, we have proposed a model, actually is a very elaborate model, which was able to explain a wide variety of motor functions and some cognitive functions of basal ganglia. And all that effort was stretched out over like more than 10 years, and which even resulted in a comprehensive book, kind of monograph. So I'll talk about some of this stuff in the next class. So let us stop here.